Hello friends, welcome to BMH Learning. In this video, we are going to talk about adsorption chromatography. So what is adsorption chromatography? Adsorption chromatography is one of the most old chromatography technique. In this technique, a mobile phase is used which can be either in liquid or gas form. The most important thing here is that adsorption of mobile phase occurs over the surface of a stationary solid phase. In other words, the mobile phase moves on the surface of stationary phase, and through the mobile phase, the components of samples to be separated also migrate. Adsorption chromatography principle. This technique involves the analytical isolation of a chemical mixture. The separation of particles relies on the interaction between adsorbate and the adsorbent. Now, what is this adsorbent? Adsorbent is a substance onto which the adsorbate is adsorbed. This will make more sense when you will know about stationary and mobile phase. The adsorbent is generally made porous in nature. The high porosity provides the adsorbent a high surface area so that it can adsorb components on its surface. The adsorption occurs mainly by intermolecular forces, that is, interactions between particular groups of both the adsorbate and the adsorbent. Commonly used adsorbents are silica gel, cellulose, alumina, etc. Now let's talk about the two phases used in adsorption chromatography. So we have the stationary phase. So this is the phase which doesn't move. And generally, stationary phase is applied on the surface of a solid support. The components in the mixture have different affinities for the adsorbent. A specific component having more affinity with adsorbent, a specific component having more affinity with the adsorbent will bind more strongly with it and hence elute later, while the component which has lower affinity with the adsorbent will bind weakly with it, thus travel faster and gets eluted earlier. The second phase is the mobile phase. This is the phase which will move. It can be either a liquid or a gas. When a liquid is used, it is referred as liquid-solid chromatography. And when a gas is used, then it's called gas-solid chromatography. This is the mobile phase through which the components to be separated move actually. So it acts like a medium for the component mixture. Now, there are different types of adsorption chromatography techniques. First is thin layer chromatography, where a thin layer of the adsorbent is applied on the surface of solid support. The adsorbent is nothing but the stationary phase. Second is column chromatography. In this type, a specific column is chosen according to the experimental need. The column is packed with the stationary phase and the mobile phase moves down the packed stationary phase. This way, the particles to be separated also move through the column. Next variant is gas-solid chromatography. This is another type of chromatographic technique which also uses the principle of adsorption. But this is specifically for those substances which have reduced solubility in the mobile phase. Adsorption chromatography applications. It's used for the separation of amino acids. It is useful in the isolation of antibiotics. It's also used in the identification of carbohydrates. It is very helpful in the isolation and identification of fats and fatty acids. It is used in separation and determination of peptides and proteins. This was all. Thanks for watching.